So, have you ever wondered how we actually validate all this amazing new health technology hitting the market? I mean, every single day there's a new wearable claiming it can track your vitals just by sitting on your wrist or scanning your finger. But how do we know they aren't just making educated guesses? How do you mathematically, scientifically prove a health wearable actually works before it ever reaches the consumer? Well, it requires a level of rigorous methodology that is absolutely fascinating. In this explainer, we're diving right into the exact methodology for ensuring medical consistency across a very specific, highly sensitive dataset. Because, as you'll see, good data isn't just passively collected, it is meticulously, purposefully engineered. We're going to deconstruct the anatomy of a clinical dataset that was used to validate a brand new, non-invasive pediatric blood glucose monitor. Okay, let's dive into this. Here's our architectural blueprint. We'll look behind the medical dataset, build out some demographic reality, set our clinical guardrails, and finally execute the dual device protocol. Starting right off with Section 1, behind the medical dataset, looking at the macro population scale. To prove a device works consistently, you really need a blueprint that is both incredibly expansive and highly controlled. You can't just test a couple of people on a random Tuesday afternoon and call it a day, right? It all begins with this number, 123. The methodology for this study brought together exactly 123 unique pediatric patients. Now, why does that matter? Well, in clinical data validation, your sample size is literally everything. Having 123 unique patients provides a robust enough sample to account for the natural biological variations among different kids, which gives the dataset the statistical power it absolutely needs to be taken seriously by the medical community. But, a big group of people is really only half the equation. You also need time. Each of those 123 patients provided a full 30 days of consecutive readings. We aren't just looking at a single snapshot in time here. We're looking at incredible longitudinal depth. Just think about it. 123 patients tested every single day for an entire month. That generates nearly 3,700 unique paired data points. And that is exactly how you build a solid foundation. Moving into phase 2, building demographic reality, where we're crafting a representative pediatric sample. Look, having 123 kids is fantastic, but if they're all exactly 10 years old, your data is pretty much completely useless for anyone who is 5 or 15. To accurately mirror the real world, researchers brilliantly segmented the pediatric age range into 4 distinct subgroups: 5 to 7 years, 8 to 11 years, 12 to 15 years, and 15 to 18 years. This lines up perfectly with standard pediatric care categorizations. The methodology ensured that the number of patients assigned to each subgroup was strictly proportional to the expected population distributions across childhood and adolescence. And to guarantee this demographic spread was mathematically flawless, the patient birth years spanned from 1992 all the way to 2020. By establishing these very specific birth years, the engineers behind this dataset ensure that the exact calculated age of the patients at the time of data collection hit that 5 to 18-year-old target perfectly. Every single patient identifier from patient 001 up to 123 is tied directly to a specific year of birth to maintain this absolute demographic reality. Okay, on to phase 3. Setting clinical guardrails, establishing those strict boundaries for realistic medical data. Now, what's really interesting about this step is that even with a perfect demographic spread, data generation requires incredibly strict structural integrity. Medical data has to look and behave like real biology. Notice the consistency in the blood glucose range here. Despite the massive developmental differences between a 5-year-old and an 18-year-old, clinical guidelines dictate a super strict normal range of 70 to 100 milligrams per deciliter across all subgroups. The methodology enforces this boundary to ensure that the data set remains firmly 100% within normal, healthy pediatric blood glucose levels. But here's the catch. You can't just cross your fingers and trust that data will magically stay within those ranges. You have to enforce it professionally. And that is exactly why having a registered nurse collect the 30 days of readings is so absolutely critical. A registered nurse guarantees clinical strictness. They ensure that the collection process is flawless, the testing environment is tightly controlled, and that the readings authentically represent those age-specific normal ranges. It adds a layer of undeniable medical authority to the whole shebang. Which brings us to the final construction phase. The dual device protocol, basically the ultimate validation face-off. We've built the demographic sample, we've set the clinical boundaries, and now we test the technology. So the crucial point here is that we are testing a brand new, non-invasive technology side by side against an established gold standard. In one corner, we have our baseline, the MD2-BLE self-monitoring blood glucose system, which carries official FDA clearance. In the other corner, we have the challenger, the RE.DOCTOR Vitals 1.6 ppg device. The methodology strictly requires that for every single data point over those 30 days, readings from both of these devices are collected simultaneously.
Okay, so we've got the theory, we've got the methodology, and we have the protocol. But what does this actually look like in practice? Let's take all of this abstract methodology and ground it into a tangible, row-by-row reality.
Let's look at how this builds a micro-narrative for just a single subject, patient 001. This is a 6-year-old child born in 2020. On day 1, the FDA-cleared MD2 glucometer reads their blood glucose at 95. At that exact same moment, the non-invasive RE.DOCTOR device reads 94.95. On day 2, the established device reads 76, and the new device reads 75.3. Day 3, 99 versus 98.61. Just notice how incredibly close the new RE.DOCTOR device tracks against the FDA-cleared standard, all while staying perfectly within that 70 to 100 clinical guardrail we talked about earlier. It's almost identical. And when you zoom out, this structured spreadsheet elegantly compiles all of these perfectly engineered data points into just 6 distinct columns. You have your unique patient ID, the year of birth, the calculated age, the specific day of the 30-day trial, the gold standard FDA-cleared reading, and the new PPG device reading. It's clean, it's organized, and it proves beyond a shadow of a doubt that the methodology for ensuring medical consistency was executed flawlessly across thousands of rows of data. By building a dataset with such strict demographic reality and clinical guardrails, we can actually confidently validate new technologies that could literally change lives. If we can validate wearable tech with this level of pediatric clinical precision, matching traditional invasive methods almost perfectly, so what is next for non-invasive health monitoring? We might be looking at a future where pain-free, continuous health tracking isn't just a cool idea, but a validated everyday reality for everyone. Definitely something to think about.